So let us now sum up the features of PBR322 plasmid. So here you are. The DNA of interest or insert may be inserted into any of the resistance genes here. So it will be inserted into tetracycline resistance gene or the ampicillin resistance gene. Whichever gene the DNA of interest is inserted into, the cell becomes susceptible to that specific antibiotic. Right? So let's assume that the insertion happens within the tetracycline resistance gene, in which case the cells that undergo successful recombinant transformation, which means that the insert has got inserted into tetracycline resistance gene, and this plasmid has transformed into the host cell, then those cells will be susceptible to tetracycline. So to select for recombinant transformants, essentially what you're looking for are cells that are ampicillin resistant but tetracycline sensitive, because our DNA of interest has got inserted into the tetracycline resistance gene. This gene is inactivated. However, ampicillin resistance is intact. So in this case, the recombinants will be ampicillin resistant and tetracycline sensitive, right? And this is a strategy that can be used to identify your recombinants. So here on top, I represent the population of cells that you get after the transformation experiment. So if you look at the sensitivity with respect to antibiotics, these cells, because they do not have a plasmid, these will be sensitive to both tetracycline and ampicillin. These cells here, which are basically transformants, but with the self-ligated plasmid, here both the tetracycline gene and the ampicillin resistance genes are intact. So these cells would be resistant to both tetracycline and ampicillin. And these ones here, the cells of our interest, the actual recombinant transformants, these would be tetracycline sensitive, because the insertion was in the tetracycline gene, disrupting the tetracycline resistance, but they would be ampicillin resistant because the gene for ampicillin resistance is intact in these plasmids here. So now the question is, how do you identify your actual true recombinant cells from the non-recombinant transformants? So first, of course, you want to eliminate the non-transformants from your population. So you simply plate the transformation mix onto a ampicillin positive plate. So these cells, because these are ampicillin sensitive, all of these cells would die off. And only cells that can survive in this plate are the non-recombinants as well as recombinants here. So you have a population of cells that are either these type of cells or they would represent these type of cells. So now I represent them with blue dots and green dots here. So these are your blue and green dots here. So these represent your non-recombinants as well as recombinants. And from here now, you want to identify the cells that are your true recombinants. So for that, you do a replica plating. And when you do your replica plating, you replicate the plate onto a ampicillin as well as tetracycline plate. So now you know that your recombinants are tetracycline sensitive. So therefore, when you do a replica plating, your recombinants will not grow here. Only the blue cells that are your non-recombinants grow here. So only the non-recombinants grow here, and the recombinant cells do not grow here. So now you can compare the two plates, and the colonies that are missing in the ampicillin positive, tetracycline positive plates, but present in the ampicillin plates are the ones that are your true recombinants. So this is a two-step based selection that you do to finally arrive at the cells that are your recombinant cells. So the colonies that grow in the ampicillin positive plate, but not in ampicillin and tetracycline positive plates, carry the actual recombinant vector. So now that we have gone through the selection process in PBR322, one major question that I want to ask you is, what are the major limitations of PBR322 and why did we need to move on to other vectors like PAC8 and PAC18? So here is your answer. First, of course, is that the selection process is slightly elaborate. And it is a two-step selection. You have to plate the cells twice to actually get to the final recombinant transformants, and that too in indirect manner. A one-step color-based assay for differentiating recombinant transform cells from the non-recombinant ones would be more convenient. So to address this limitation, we moved on to PAC8 and PAC18, which can basically give you a color-based selection, and this process is known as blue-white screening. We'll see this in details as we move along, and we'll also go into the molecular basis of blue-white screening. Then, of course, the second important limitation with almost all plasmids is the maximum insert size that they can hold. So not more than 5 to 6 kb of insert can be cloned into PBR322 or other plasmids, and that is a major concern.